when David was actually just finishing up as a graduate student in George Fayer's lab, working on photosynthesis. He actually had read my '82 paper and gotten intrigued by the relation between that and what you could do with a simple physical model and neurobiology, and then went off and said, I want to do neurobiology. And so went to David Tank's lab as a postdoc. So I've known David for a long, long time. Often argumentative, but very well. I'm going to start off by trying to say something about the different kinds of theory. Many of you come from physics, many of you don't come from physics. And I just want to remind you of the different roles of theory, because in the longer run, in the rest of the talk, I'm going to talk about a particular kind of theory. I don't want to present it as the only way to do the theory of neurobiology, or even as necessarily being correct. But I believe it's the kind of theory which is one of the essential ingredients. I want to illustrate that idea by some things which come from physics and understanding how theory went in physics. If you look at the theory of superconductivity in physics, well, superconductivity was experimentally discovered in roughly 1908. I may have the year wrong slightly. It was 20 years before quantum mechanics was formulated in such a way that you possibly could have a theory of superconductivity. It was another 30 years before the BCS theory of superconductivity actually took the mathematical equations which you could have written down in 1927 and solved them adequately to understand where superconductivity came from. Now, at the same time that it did that, it didn't do it in any real detail. It was a marvelous, marvelous theory of Bardeen, Cooper, and Schriever, which came out in 1957. But all the details of the particular material were encompassed in a couple of parameters. The superconductivity really arose at a microscopic level from the detailed interactions of electrons and phonons and from the Coulomb interactions between the electrons. And yet all those had been squished, thrown away, one particular parameter which characterized the material. And in terms of that one parameter, there were a whole host of quantitative predictions you could make relating one set of experiments to another set of experiments. But there was really no satisfactory, truly microscopic theory, because everything had been aggregated onto a collective parameter which represented, in some sense, the mean value of all kinds of distribution of parameters, all kinds of details. And it wasn't until another 15 to 20 years went by that you could actually calculate what materials ought to be superconducting by getting down to the fundamentals of all the interactions. Prior to BCS theory, there was another theory, the Landau-Ginzburg theory, which is a macroscopic theory of superconductivity, relating all kinds of macroscopic properties of the system without any understanding of the microscopics whatsoever. And that theory was also very useful. And all these levels of theory can go on more or less independently. And you have to have a tolerance for these. I mentioned the word tolerance because as I've grown older in science, there's an adage, friends may come and friends may go, but enemies accumulate. And you do have to have a tolerance for the way that different people choose to define and work on problems. Now, when you turn to biology, there are additional problems as difficult as a physicist. One is that the most important theory in biology is undoubtedly Darwinism, which has no mathematical content and no quantitative predictions whatsoever. And it wasn't until we were able to go further than that and say, well, inheritance, then Mendel and classical genetics came along. Mendel and classical genetics were beautifully developed. There's nice mathematical content there, some quantitative predictions. But that was done without any knowledge of what the genetic material was. And so it wasn't true, as people often claim, that you must know all the microscopics in order to understand the higher level of things going on. And in fact, one didn't really understand what a gene was until sometime around 1960. And yet, classical genetics was a very well developed and very well mathematized structure. If you turn to neurobiology, I suppose in many ways the most important theory in neurobiology, the most important mathematical theory, was the Hodgkin-Huxley theory of action potentials and action potential propagation. Now, in many ways, that is a beautiful, beautiful engineering summary of how action potentials can occur and propagate. On the other hand, it is an engineering approximation. The parameters which they actually have in there are really, once again, like the superconductivity case, they're averages over all kinds of somewhat related things. And if you go outside the range at which the equations were established experimentally, then the equations break down. Not astonishing, but it just tells you that the equations aren't fundamental. The equations are an engineering approximation. And in the way of trying to understand what are the equations capturing, biologists were not particularly good at trying to say, well, what are they actually capturing versus trying to understand the equations for understanding all the bells and whistles. Years ago, I went to a talk by Chris Zeeman, who is best known for his work in what's called catastrophe theory in mathematics. The talk was entitled something like Heartbeat and Nerve Impulse. And what he was trying to do was to describe how it is from the nature of heartbeat and the nature of nerve impulse, you could understand how many first-order nonlinear differential equations you would need in order to be able to account for them. I thought he did a brilliant job of trying to describe the mathematics of the difference and to understand there were these classes of behavior, and you could be in one class or the other class, and either of the classes could have all kinds of things instantiating how they operated underneath. Hodgkin happens to be in the audience, and Hodgkin's reaction to somebody trying to understand the mathematical structure of the equations was, love too has something to do with the heart, he said, and sat down again. Now, what I'm going to really try to describe has to do with the following. If I look at large physical systems, air in the room, for example, all large physical systems are made of simple components, have collective properties, emergent properties, which are consequences of their microscopic behaviors, but rise above them. Look, the velocity of sound propagation in the air, comes as a result of these details of the molecular collisions. Without molecular collisions, there would be no sound waves in the air. And yet, we describe the sound waves in such a way that it's fluid dynamics. There are no molecules in the way we describe sound waves to freshmen, and we give an accurate account of the sound waves to them. And the Navier-Stokes equations are just a classic example of there's really microscopics underneath. There's a theory which relates the microscopics to the macroscopics, but the equations which are macroscopic are the ones which are very interesting to try to then understand the world with. Now look at psychology. Psychology is a science of macroscopic behavior. Different animals have different psychologies or different ethologies. All of these behaviors are macroscopic. And in many ways, you could say, insofar as psychology exists as a science and a quantitative understanding of this person is going to behave roughly like that person because of the same species, you know that the brains of this person and that person are not identical. They've got different numbers of neurons in them. Even things as elementary as the number of cells on the retina can be different 10% from one person to another. There are no identified cells in you that you can identify as the same cells in me. And yet our psychologies are in some sense the same. Our psychologies have got to be the emergent properties of this large network of neurons. And so I look at one of the things that understanding neurobiology has got to do is to understand how the macroscopic laws which we attribute to behavior arise from the underlying microscopic details of the neurobiology. And that way they're quite different from understanding how computer macroscopic properties come from computer details, because at least in old-fashioned computers, every computer of the same type was built exactly the same way, had the same number of transistors in it, same number of logical circuits. The whole thing was isomorphic. This computer acted like this computer because of the one-to-one correspondence between the microscopics. And that's just not true in psychology. Two people behave in ways that are similar, for ways which don't follow from the, look, they're the consequences of the underlying microscopics, But the isomorphism of the behavior of this person does not come from the isomorphism of the details down at the bottom level of neurobiology. So I'm going to talk for the rest of the talk today about trying to get from microscopics to macroscopics. Now, is this a reasonable time in the history of neurobiology to try to do such a thing? Quite possibly not. At one point I got very interested in consciousnsness, and I asked a bunch of my betters their views on consciousness as a subject of science. I asked Feynman, Minsky, Bill Anderson, Francis Crick. Francis Crick actually gave the most interesting answer. Francis Crick, when I asked him about consciousness, went off on a diatribe about Mendel and the peas, a diatribe relating to the fact that as soon as there was Mendelism and inheritance, you knew that there was something fundamental about inheritance which you didn't understand. The notion of a gene began to develop. It took 100 years to develop, but it was so much the essence of inheritance, and inheritance was so much the essence of biology, that you had to study it, had to begin studying it, even when you didn't even have all the tools necessary for it. And in that same, and one of the reasons for doing so is that such a study informs the kind of tools, the kind of microscopics that you look for. And so the reason I'm going to present for doing what I'm doing now today is that although it's too early to do it, trying to do it with some kind of responsibility will inform experimentalists about what kinds of things they might do in more detailed neuroscience. So what kind of overall subjects might I be interested in this regard? Take perception. I'm not going to spend much time on perception, but let me just see if I can wake up the machine. And when you look out in the room, you don't see a map of intensities of various colors. When you look out in the room, look at something like this. You don't have an intensity map. You actually have, you see an object. You see a green cylinder sitting on a checkerboard. You see black squares and white squares. This is an artificial generated thing, but in general, you look out, you don't have a bitmap image of a picture. You see the world as objects. And that's very robust. And you even see it as objects so robustly that you're willing to call black white in order to see an object. For example, those two squares obviously have the same intensity to those two little fractions. Yet one of them is taken from square A and one of them is taken from square B. Just mask it off. You call one black, you call one white, just because you're so determined to make objects that you recognize out of this pattern. So you don't have a bitmap image. And data isn't always simple to interpret. Things like this, there are multiple interpretations. People look at this and see two opposing faces, or a vase is a well-known illustration from psychology, but it just illustrates the fact that you try to take these intensity maps and make something familiar, something object-like out of them. And let's just remind you that the things which you will say are the same object, the same face, for example, any of these three will remind you of Abraham Lincoln. That one at the top left, you have to squint to see it. From Bailey Eulis's work, you squint, it blurs out all the artificial edges there, and suddenly you see the face of Abraham Lincoln. But stimuli which are enormously different are said to be the same object. And the objectness is really a very collective property of an immense array of sensors which have sensed the environment. But the object is a collective idea. You have the same kind of collective things happening at the motor side. You train a rat to hear a buzzer and press a button, and Hebb writes, "A rat trained to depress a lever to get food after a tone sounds may do so from any of several positions. He may climb on the lever, press it down with his left forefoot or his right, use his teeth. Very often all that can be predicted after the response is learned is that the lever will move downward." So you don't in learning systems learn simple things which are this muscle twitches because you hear the bell. Higher animals learning things learn intentional motions. They learn something which is much more macroscopic than a simple sort of reflex arc. And the kind of the behavior Hebb was describing is an output example of the kind of collective thing you can see here in objects in the input side. Now, the thing which I would like to deal with is thinking. And I'll give a rough definition at the moment. Thinking allows an animal to make an effective action in a novel situation based on a mental exploration of possibilities and previous knowledge. Thinking, taking an action in a novel situation based on previous knowledge. Now, if you all have ideas about thinking, and the way you think when you play chess, for example, you do exactly the kind of thing I've been describing. The situation isn't quite like any situation you've seen before, and so you think about, oh, if I move here, my opponent will move there, and so on. That is a kind of mental exploration. But if I'm going to make any progress, I can't look at thinking which is done consciously, because I have no idea what consciousness or conscious awareness is. And so it's very important, if you're going to do anything about thinking, that you try to work on thinking in such a way that it doesn't require conscious awareness. Now, can you think without conscious awareness? Psychologists will, I think, argue that you can, and they actually try to study such things by studying what they would call insight, or an aha moment, when suddenly something gels and you don't know quite how it came together, but you suddenly know that something is right. Here's a simple kind of thing that a psychologist might do. Here are 3 words. Find another word which goes just hand in glove with each of these 3 words, either as a compound or just used together with it. And you can think about this consciously, or you can just look at those words and your mind drift off somewhere. And the psychologists like to study what happens to those people whose minds drift off somewhere, and then suddenly they know the answer. Because in that case, you've been thinking without conscious awareness, and that's the kind of thing which I might be able to get my teeth into. Everybody understand what this problem is? Anybody have a word that goes with those 3? Pardon? Yep. Here's another one. Fire, yeah. Here's one. Post, beautiful. But the psychologists can ask when people have found these whether they found them by considering word after word after word or non-consciously, and they can look at EEGs from one set of instances or the other and really try to get a little bit into the details of how you think in a non-conscious fashion. But nevertheless, I just want to put these up as illustrations of being able to think perhaps non-consciously. Now, people in neurobiology, by and large, do not study thinking. Why not? Historically, of course, there's been the single cell aspect of neurobiology. You can record from single cells, and multiple cellular recording has only come much more recently. And most of the dominant experimental paradigms have been established for single cell recording, and the same experimental paradigms tend to be used even when you can record from multiple cells. And the difficulty with thinking as such a subject is that if thinking involves solving a novel problem, you have one shot You can't try to average a whole bunch of occurrences. The second time this problem is given to you, you've already solved it once. You can't average the data into the first set of data. You have to look at thinking as a single-shot process, and yet, and it might be quite different in one person and another person, and yet you have to do the science in such a way that the science is reproducible. Otherwise it's just not science. And to make life worse, of course, you have the fact that there's the variability problem in neurobiology. These are experiments from Dima Rindberg, looking at neurons in the olfactory bulb with an anesthetized cat, and you can see that with this particular neuron, mitral cell, is quite interested in one odorant and very uninterested in another odorant, and you can say, I can begin to understand how I can distinguish amylacetate from citral on the basis of behavior of neurons like that. That's obviously several different tries. The response is fairly reliable. Shouldn't be hard to build something which functions from that kind of inputs. If, on the other hand, you ask the cat to do the rat to do a task, and the rat isn't anesthetized, you wind up with traces which look like this, in which suddenly the variations between what happens with amylacetate are as great as the difference between amylacetate and citral. There's a huge amount of ongoing activity in the absence of the odorant. And look, the animal is alert. Mental things are going on. You're not just stimulus dominated. But once you admit that things are going on and you're not just stimulus dominated, it becomes much more difficult to control experiments in such a way that they're reproducible. Clearly, you might hope to have a single cell description of how odorants are discriminated if you only had data of this sort. If you have data of this sort, it's almost certain that you must have a population of cells that you simultaneously record from before you'll be able to understand how the robust recognition takes place when you absolutely have destroyed what seems to be the faithfulness which you could have had in a cell. All right. So without further ado, I'm going to head now into trying to with this definition of thinking, what can I do as an experimental paradigm so that you will understand, yes, it's thinking, and yet it's an experimental paradigm which you could apply to a rat and try to make progress studying the rat and thinking about such things. So the illustration I'll give of thinking is of the following sort. You're on the university campus that you know best. You just parked the car and you're halfway to your office. Somebody stops you and asks you how you get to the library. Now, you've probably never actually gone to the library from that particular point. On the other hand, you know the campus well. You look at the person and say, as a visitor, I better pick a simple route. Let's not that route. Let's tell them about this route. And you mentally explore a couple possibilities for what you'll say to this visitor, pick one of them, and describe it. So you've solved a novel problem on the basis of knowledge that you have. Now you can ask a rat exactly this same kind of question. Supposing I have a maze. This is a sort of a maze. It's not quite a maze because it's been tailored in such a way that it has different closed segments which don't connect out to other places. And as a maze, I would want to have all kinds of decorations all along the walls of it, so that if you looked, you could tell where you were in that maze on the basis of this decoration here and the decoration over here are just fundamentally different. So any particular point is identifiable. Supposing you have a maze of this sort, and you insert a rat through a trapdoor in one place or another, and you do this day after day, the rat will go on and explore the maze, and after a while will know the details of how the maze is laid out. So this is the previous knowledge. Let the rat wander around with no motivation other than what you might call curiosity. Now, on the next day, you make the rat very very thirsty, and you insert the rat here where there happens to be, you've placed some water, and now you take the rat out after having let it lap up a little bit of water, and put the rat in another spot in the maze. The rat knows the maze. The rat ought to be able to ask itself the following question: can I, by wandering around, find the water, or am I not in the section of the maze which actually contains the water? Now, the rat could do this by physical exploration, but the rat also has the potential for doing this by mental exploration. That's the way you'd solve the task, and there's nothing in what one knows about rats which would keep them from trying to do such a mental exploration. And what you'd like to have is an experimental paradigm in which you can actually examine this possibility. And there are two things which the rat might do. First of all, the rat might have a mental exploration in which it actually found out it's not worthwhile looking at all because there's definitely not water available to me where I am or in the pathways I can get from here. Or it might have a way of saying, water is available, I can figure out how to get to it, and in one shot I can now get to it reliably because I've understood by mentally exploring what the trajectory is I should follow. I don't have to follow a random walk physical trajectory. I can already have done that mentally. So this is a kind of experiment which you can clearly do. And people used to do this kind of experiment in a more simple sense in rats back in about 1950. You would have, this would be a very simple maze, simply a T. The rat would explore this system with no rewards, but the floor is different on the two sides, the boxes are different at the two ends, and so on. But there's clear things differentiating where you are along here. And then the rat would be taken off somewhere, this box would be disconnected, the rat would be taken off, put with this box and some water over here, and then let the drink for a moment, and then take the rat out of that box, hook the box back up, wait 15 minutes, put the rat here, and you can now ask, does the rat understand that you go this way to get to the box, or does it actually have to do a random exploration in order to find out where the water is? And this kind of experiment, the vast majority of the rats actually are capable of solving this problem. Immediately they start from here and make the correct turn to go to the box which had water in it, although the water was never in it before, other than when the box was isolated. Now this is such a simple experiment that you can't tell really very much about mental exploration, but what I've described has been simply an elaboration of this experimental scheme already in place by the psychologists studying the field which is known as latent learning. And you can do single trial experiments, and one of the nice things about thinking of a maze of this sort is you could do many different single trial experiments of finding water somewhere from a different starting point after the rat once learned the maze. So there would be a way of trying to use a rat which you had operated on many times before you had to discard the rat. So although each experiment on mental exploration is a single experiment, yet you could use a rat over and over again and use the fact that the rat had learned this maze many times. So there's my definition of mental exploration. And the thing which I think makes it approachable from the point of view of neurobiology is in the kind of situation which I've been described here. You ought to be able to see, by looking at by looking in detail at a many neuron view of what is going on before the action is taken, you ought to be able to see that you can understand why that particular action was taken when the animal didn't take it. In short, if the mental exploration should wind up with a solution to the problem which is in some sense recapitulated when you actually take the physical action. All right, I'm now going to construct a neural network which has an emergent property that it is able to do this kind of mental exploration, able to solve this, in principle, this kind of problem. It's based very, very, very roughly on the hippocampus, of which you have two typical structures, typical old cortical structures. And the salient point of many of the cells in the hippocampus is the rat knows an environment, because suppose the rat knows there's some kind of environment, which I've drawn as rectangular here, and there are different, there's just walls, color, texture, designs, and so on. So you know where you are in that environment. And as you wander, as the rat wanders around in that environment, typical hippocampal cells become place sensitive. That is, I'll have a cell, for example, which fires strongly when the rat is here, fires less strongly when the rat is nearby, less strongly, and fires not at all in the rest of the space. These are called place cells, and people have understood that there are place cells in the hippocampus. These place cells are learned. That's been part of the hippocampal lore for a long, long time. Yes?
Is there evidence when most of the place cell literature is the animal running on a maze with multiple arms, or an animal moving within a closed container? You had something a little more complicated.
People have not really looked for place cells in more complicated systems that I know of. And pardon?
That's the first experiment.
Yeah.
The question is the first. Is to read literature from 60 years ago, and the rat's back in the game.
Yeah.
If you could place that.
And so much of the place cell literature is based on also exploring basically a flat terrain. You think of one of these palm tree rats, which lives up from the palm fronds and runs down, has an area down on the ground. Topologically, it's nothing like a plane, and yet I would wager that place cells would exist there too. For anybody who's able to do telemetering recording, it would be an interesting experiment. But I'm going to presume that place cells have these classic properties which have been deduced, as David points out, only in relatively simple learning situations. And this just shows, what I showed you before was a cartoon of what place cells look like. This just shows recordings from many different neurons in hippocampus, showing that you get place fields which are often single spots, sometimes two spots. But each cell has its own different characteristic, and typically the cells respond to no, 0 or 1, sometimes 2 locations. Now, one of the unusual features of the hippocampus in this regard is that place cells, supposing I record from a bunch of place cells in the hippocampus, place cells which are adjacent to each other don't have any preference for having activities which correspond, having chosen centers of activity which correspond to close physical locations. So it's almost as though if you are at a particular point in physical space and recorded from the cells that I've shown you, the cells which are actually active when you are at this point in physical space are distributed all over the hippocampus. And in order to make sense out of the system, I want to actually just make displays which are different. I'll make displays which are different by mapping all the cells which are mapping the cells in the hippocampus to a display where the activity of a cell is displayed at the physical position that the animal is in for the maximum of the cell response. That is to say, when I wander around the hippocampus, what I would actually see in the hippocampal recordings is a flickering response of look like this. But if you actually just redo the map a bit, the same activity pattern, which would just flicker around in an incomprehensible fashion when the animal moves around, once you redo the map, when the animal's here, I have this set of cells which are active. The animal moves over here, there'll be another set of cells which are active. There'll be a clump of activity in this mapping which takes place and moves around as the animal moves around. Yes?
So what is the correlation length sort of associated with it? Is it some fraction of the visual field or more visual field?
This will do. I think what you're asking is, what is the fractional size of this blob compared to the size of the environment? Is that basically what you're asking? And it's a few percent.
So, and that environment is supposed to be what? So that's
This environment would be the animal was enclosed in a box, which is basically, say, a meter by a meter. And wandering around in that environment, there are visual clues all over the walls, and this would be the responsive pattern of the cell. Yeah?
Are the flash points the same?
No, there's no correspondence between. Each rat will learn place cells, but whether a particular cell will be what a particular cell will respond to in one rat, there's no way of even identifying a particular cell in the other rat that would correspond to it. Both rats will have place cells, and they will both be distributed. In each case, you'll have to redo this map. It will be quite rat specific, but once you redo the map, you ought to be able to see the blob of activity wander around when the rat moves around. And in this, when the rat is here, you get some kind of pattern of activity, and the real hippocampus would look like that. When the rat moves over here, in the hippocampus to where water is, there'll be a set of neurons which will be active in this location. They'll be distributed throughout the hippocampus. But if I connect that set to something which says there's water over there, then I'd be able to make a simple system which, when the rat was over here, drove a cell, because all the place cells which corresponded to this location would be simultaneously active. When the rat was elsewhere, that cell wouldn't get driven. So it'd be easy to make a recognition system in which you recognize when the clump of activity was at some particular location. If the rat were doing physical exploration, that would be a way that the rat could say, oh, I'm near this place where water is. I'm beginning to drive that cell over there. What you have to do mental exploration is to have a clump of activity of that sort which is driven, which wanders around without the animal itself being active. Say just one little more thing about place cells. One of the interesting things coming from the Burgess and O'Keefe laboratories is the fact that you can have an animal which knows, say, two different environments, called one of them rectangle, a square environment and a circular environment. And typical place cells either have a place cell in one environment and not the other, or have place cells in both environments which don't correspond. But you can know two environments simultaneously, and there's overlap between these systems. Some of the place cells are special for one environment, some for another environment. Others respond to both. And one of the interesting things which they find is that in their hands, if you start making a confused environment, which is somewhat like one and somewhat like the other, the place cells belonging to one environment will be active, or the place cells corresponding to the other environment will be active. There seems to be something which is like a phase transition between I believe I'm in one environment or I believe I'm in the other environment. But I put up both of these at the same time here to see, so that you can see that you can know two different environments can involve the same place cells, and so you can think of this as knowing two different sections of that maze that I drew up earlier. All right. So supposing I have two different environments here, I call it a rectangle and an H environment. There are two different ways I can map a given cell as a place cell at different places in the two environments. So there are two different ways I can do a mapping. And in one way, if I do the mapping corresponding to the R environment and the animal is really in the R environment, then I'll get a clump which moves around when the animal moves around. If I do a mapping which corresponds to the H environment when the animal is in the R environment, and the animal moves around in the R environment, I'll get patterns like this which never produce blobs. And the same is true in the other direction. In short, you can understand how, in terms of the sort of blobbiness of response, once you make the right kind of mapping, an animal moves around in an environment and a corresponding blob moves around in excitation space, properly represented. And if I try to make the blob move, if I use the wrong transformation, use the transformation which corresponds to the wrong environment, I don't get a blob. And on this basis, you can begin to see how you can just tell the difference between when you're in one environment and when you're in the other environment. All right. So much for place cells. What can you do with them? Well, if you're an animal, we're trying to do physical exploration, looking for water. You would start from somewhere and just wander around in an enclosed space, hitting the walls. This has just been generated as a random walk with memory, with reflections. But you can see a simple physical trajectory pretty soon would explore much of space. So that's a kind of exploration of physical space that you'd like to have possible for mental exploration, except what you have to do is to do it mentally, rather than have the animal move around. You have to have neural activity move around in such a way that it would be doing the same moving around if the animal were just thinking about it. So what you have to be able to do is to mentally explore extensively to search for water. And the only way you can mentally explore is by making a stable activity clump and having that activity clump move. Now, it's easy enough to make a stable activity clump because if you start working with elementary versions of neural dynamics, there's a slight lack of correspondence between my screen and that, which somebody who's really familiar with laptops can get around. What do I do on the display? There's excuse me? Okay. Big enough? Big enough, okay. Let me just, yeah, okay. So supposing you start with some basic, simple artificial neural network equations. You have an input current to a given cell. There's a term which comes from a synaptic current and the firing rate of other cells, and then there's the term which comes from the duration of the synaptic current. So these kind of basic equations of very simple neural networks. The S's represent the strengths of connections between the different neurons, strengths of synaptic connections. The signs of the S's determine whether it's an excitatory or inhibitory connection. And the tau synapse, which occurs here, is the lifetime, taken to be a simple exponential, of a synaptic current. And the input-output relation of a neuron is some simple sigmoid function. And I'm going to take a network in which there are two terms in the connections. One is very explicitly excitatory connections, which are learned, and the other are weaker inhibitory connections, which are equivalent to a global inhibition system. And if the connection matrix is symmetric, there's a Lyapunov function behind this, and the determinant and the structure of this is that there's some kind of attractor dynamics, and that all the activity will relax to an attractor. Now, supposing you do explorations in one dimension, and you use a rule which says neurons which are on together, which fire together, wire together, the typical Hebbian synaptic kind of thing. I'm going to arrange the neurons for simplicity as place cells in order in one dimension. And the cells which are active together are the cells which are close to each other. And if you learn a synaptic connection matrix on that basis, it'll basically be a diagonal stripe of some width. I killed the connections on the diagonal. I have no cell connections. That's more or less material. But that's the kind of connection pattern you'd get. And if you had a one-dimensional system of that sort, there would be it would produce a line attractor. There would be the energy would be independent of where you put that bump of activity. The bump of activity could be placed anywhere. It would be stable. You're beginning to have something which would be useful for mental exploration, except for the fact that the bump is stable anywhere. Now, supposing I want to learn another environment. Well, remember I told you that being a place cell in one environment has very little to do with where you are in another environment. So we can think of the other environment as having some arbitrarily different way of scrambling the neuron locations. And so that if I take that synaptic matrix with an arbitrary scrambling, it produces something like this. And in terms of that arbitrary scrambling, I can now learn another environment which looks like this, and you realize that this is just a question of putting in multiple memories. You would embed multiple linear attractors. There's a little bit of crosstalk, but you can embed multiple linear attractors in such a network. And you can go on and embed multiple planar attractors in networks in two dimensions. Same idea, no difficulty doing it. Now, we're interested in mental exploration. If we go to two dimensions and try to see, we want to have a mental exploration in which we wander around in this sort of fashion. But if we go to a system which knows multiple two-dimensional environments, multiple planar environments, and then say, let's start somewhere, how does the system evolve? The activity pattern will produce a bump somewhere, and that bump will move a little bit, and then that bump will stop. These green dots show the locations at which I've started the dynamics. The red dots show the places it ends up. Of course, it's simple attractor dynamics, and you start somewhere and you go somewhere. There's finally a bottom of a valley, and you stop. This kind of motion in time does not produce exploration of the plane. And you have to produce exploration of the plane if you're going to have mental extended activity exploring and looking for things. So what can you do about it? Well, most neurons in cortex, if you put it... Yeah. Yeah. Yeah.
So there's no sensory input.
No sensory input. I've learned 5 planes in this particular case by using sensory input to move this bump of activity around. As a result of that, I've made a set of connections. The set of connections now has stable clump behavior. And I look and now see, I see if I initiate a clump somewhere, where does the clump move to?
And why does it stop? I mean, it's stable because you don't have any noise in the...
If there were noise, it would jitter around, but it basically stops because there's a Lyapunov function and it goes to the bottom of a valley. It would be a plane.
Then why does it move at all?
It would be because I tried to put 5 planes in, there's crosstalk, and so for anything which is supposed to be a plane, it's a little bit wavy, and so it moves, I started somewhere and it moves over to a local minimum, which isn't very deep, and it just sort of stops. And so it moves, but doesn't succeed in extensive exploration. You have to show something where you'll have continuing exploration if you want to think. Yeah. Oh, that's right. And so in this particular picture, I start in one environment, and the bump stays in that environment and moves over and stops. If I started in another environment, in this representation, there wouldn't have been a bump. So the bump is very dependent on what representation. If you started the bump in one representation, if you start believing you're in one environment, you stay doing an exploration in that environment, but you don't go very far. No. This is what happens if you're in one particular environment, and I've initiated a clump here in this environment, and that clump has moved over there and stopped. Now, if I had initiated the clump in another environment, in this picture there wouldn't have been a clump because that whole thing is mapping dependent. In the other environment representation, there would have been a clump, and it would have had the same kind of behavior. Yeah.
So if you just have one environment with that translation invariance, and you guarantee a manifold of this one. But when you add the other environments, for any one environment, you lose that. Right? So
you lose that, and so sort of instead of having a plane, you actually now have a large number of very shallow minima. And so you start somewhere and you move over somewhere and you get stuck.
How do you guarantee that you have a large number of minima that's sort of uniformly tiled?
Oh, is it? I'm just telling you now what happens. This is a picture of what happens. I start somewhere and I move over a little bit. I typically start somewhere, don't move too far, but some of the times I move a considerable distance. This is the reality of what happens. I do not have a mathematical theory of exactly how this happens.
How does this behavior depend on the number of environments that you
In this particular system, there were, on the scale of 2,000 neurons, each clump involved the activity of about 50 neurons moving around. If I put another 10 environments, if I put 5 environments in, one environment will give a plane, go as far as 10 or 15, and at that point, you suddenly are unable to stay thinking about one environment. You start jumping from environment to environment. The system loses its way. So it's just there are only a certain number of synapses. There's only a certain amount of information you can embed in them. But this stinks as a way of doing mental exploration. However, most neurons in neurobiology actually, if you have input currents which look like this or like this or like this, the firing rate goes up and then fades off. Well, most neurons show adaptation. Most synapses show adaptation. And so the model I have had of neurobiology failing to do mental exploration is obviously the wrong model. It doesn't connect with what the real neurobiology looks like. Let's just ask what happens if you put adaptation into the same picture I've had before. So if I take neurons which adapt, how do I put adaptation in? Simplest way of putting adaptation in is the simplest way, which makes the system adapt, is equivalent to making a self-synapse which is inhibitory and slow. That makes the system look as though it's adapting. And so if I do that, if I make a self-synapse, if that self-synapse has the same time constant as the excitation synapses, then I still have a symmetric network, same old dynamics, and there's the alpha null functions, and there's nothing interesting which has happened. If, on the other hand, the self-synapse is slow, then there's a whole new set of dynamical phenomena which emerge. And so it's very important in what I'm describing here that adaptation be on a time scale which is different from the synaptic scale. But you can see what happens now in one dimension. In one dimension, if I don't have adaptation, I have a flat energy function, and a bump is stable anywhere. In one dimension, with adaptation, if I try to put a bump here like this, then these neurons are going to adapt. They're going to try to drive others which have not adapted, which are actually going to be energetically more favorable in some sense. What happens is the symmetry is broken, and this initial bump will, as a result of symmetry breaking, will rush off in one direction or rush off in the other direction with fixed velocity. And what really is happening is if I have a system which, think of a bump of activity moving along, there's no energy function quite because of adaptation, but the neurons which are ahead of the bump haven't adapted yet. The ones which are behind the bump have adapted. And so it really is almost equivalent to making an energy function which is chewing things up behind you and making it always more, leaving it favorable for you to go ahead, but you've left behind a trail which says don't go back that way. This system propagates with fixed velocity in one dimension for conceptually the same reasons as action potential propagates with fixed velocity in a neuron. It just, you need that other equation of motion, which is provided in this case by adaptation. So if you put, now the same is true, of course, in 3 dimensions, and you two dimensions, and so you tend to move out of the place that you were and away from it in the direction which is continuing to move farthest away from the things you were a while ago. And so if you initiate, once again now, take a system which actually knows 2 different environments. I can initiate a clump here or initiate a clump here. And in either case, you can see what actually happened. I now plotted the trajectory of the clump as it moves around, or the trajectory of the clump as it moves around. The clump does exploration of one space, stays within that space, doesn't move, but carries out an extensive exploration now because of the adaptation, which you couldn't do in the absence of adaptation. Yeah. No, if I'm in the H, if I initiate a local bump in the H environment, that bump stays within the H representation as a bump. If I initiate within the other environment as a bump, I stay within that. And so what looks like a bump moving in one representation always looks like just a bewildering random activity in the other representation. So it's very important to have the representation as you try to plot these data. But you can see that if you are in an environment and you initiate mental exploration from that environment, you will stay exploring that environment. So that's the first part of successful mental exploration. I can explore an environment, and now if water was present in, say, at this spot in environment H, and I initiated here, I'd eventually find water. And if water was present in that, and I initiated in the other environment, I would never get an activity pattern which corresponded to a clump in this location, and thus would never identify it myself as having explored and found water.
So the electrophysiological prediction is that you're replaying the sequence of bumps that you would record as the animal did the task.
Did the physical exploration. Of course, you don't know what physical, you don't have a correspondence there yet, because you haven't seen any physical exploration. As I said, the animal, but you'd expect the animal to do this, in this representation, to propagate a bump and to find a target. And then what the animal has to do next is to actually recapitulate that and connect it to physical motion. Because if you can't do that, it's not any good.
Right. Now this example is a single path. And in a maze, you butt up against it. And what happens in these models?
I'll show you a little about what happens when the things are constricted a little later.
But the fundamental assumption here is that this mental exploration is as if, it's not virtual. It's
I'll talk at the very end about how to try to do this with more possibilities at once. At the moment, I'm trying to do it with one possibility at once. One path. If that path doesn't work, you'll try another path. When you're playing chess, how many simultaneous things are you trying to work out? One.
That's why I don't play.
Me too. Yeah. Yes, yes. Because adaptation only lasts a certain length of time, and so that as I come by here, well, I could cross where another trajectory was. I tend not to go follow the other trajectory because that stuff is still more residual adaptation. But trajectories can cross, and they can easily cross when they're a long time distance apart. But there is history to this, and so when you come back to the same point, you're not coming back to this. You're just coming back to the clump being at the same point. You're not coming back to the same activity point.
And the time scale of adaptation is the one where you go from one trajectory to another?
The time scale for adaptation basically determines the velocity with which you move along this path. Yeah.
I mean, according to the definition of thinking, it seems to me what you need to do is you need to compute the cost for all your alternatives.
You need to compute
Do the cost function for all your alternatives and then choose the one with the best.
Look, if you want to do very, very fancy computing, that's what you want to do. If you're a simple animal, what you're actually asking after is the question, can I actually find water? And the cost function is 0 if I don't find water, and yes, and 1 if I find water. And so I'm trying to keep the cost, the fancy things of choice out of this, and just say, can I actually accomplish the task, and can I find a means of accomplishing the task, and can I actually convert that accomplishing the task into a physical action?
So this traveling bump of neural activity is evaluating the cost function whether you get to the target or not.
The only time there's anything interesting happening, I go along here, and if I find the target, I will then say, I found water, and the system will then be activated to try to recapitulate this pathway mentally, but at the same time follow it physically. So it isn't a question of trying to evaluate the relative value of different goals. That's far beyond what I can do. Yeah.
So if you do end up finding water,
Yeah.
do you expect that the animal will follow exactly the same path?
In the simplest form of this, the animal would follow the same path. Now, the animal has a whole lot of systems which my model animal doesn't. For example, it understands something about orientation in the physical world. My animal doesn't have any knowledge of that. It just has knowledge of what the place cells are telling it. And so I would in this vanilla model, you would say, if I find water by some path, I will then take that same path. I have no knowledge what else to do. But the problem is to get the system now to recapitulate and to follow the path which it's followed mentally. So how can you mentally learn? How can you learn to recapitulate a trajectory? Supposing I have a trajectory and after a while it's found water. How, when I start from the same origin, could I take that same trajectory again? And I think the key to that is just a simple kind of synaptic learning. I have a whole set of connections already made. If I take the animal through a path, then there's cells which are active at the same time other cells are active as a result of those cells having place field centers which are near to each other along the path. And those cells will have an opportunity to be synaptically more connected. And so if you actually just use a sort of a Hebbian learning to temporarily bump up the activity of synapses which were active when I took a path, the next time I start from the same location, I will tend to take that same path again. And this way you can take a flat terrain, this is XY terrain, and carry the animal through a path. I've carried the animal through this sort of sigmoid, the sine wave segment of a path. And at the same time, the sensory system is telling the animal where it is, so certain place cells are active. I've enhanced the connections by about 50% of the active place cells while I carried it through the trajectory. And the next time I put the animal there and let it go, follow a mental trajectory. The mental trajectory then followed the physical trajectory through which the learning had taken place. That is to say, once the animal goes through a physical trajectory, it's easy to make connections so that the mental trajectory will follow that physical trajectory. And the same thing allows you, having made a mental trajectory which succeeded in doing something or other, to enhance the synapses in such a way that you can recapitulate that mental trajectory.
So there's no need for any dynamics to that learning rule. The reason that it works is because cells with nearby the place fields are broad enough.
That's right.
When the animal's at one location, this neuron's responding a lot, but the neuron that actually prefers the nearby place field is responding a little bit. That's enough to strengthen that connection.
That strengthens that connection. Just along that pathway, the neurons which are a long way away aren't, and those aren't strengthened. And so just the connections, which basically, if you had only one environment that you knew, you're taking a flat environment and you're embedding a little groove in that environment, and the system will then choose to move along that groove rather than anywhere else on the flat. Now, I think that it's important to carry this kind of idea all the way to behavior. Being able to recapitulate a mental exploration doesn't, I think, have behavioral consequences unless you can recapitulate it in such a fashion that it causes physical behavior. That is to say, what you really want to do is to say the animal does a mental exploration and then it does a physical exploration and see that there are neural correlates of the mental exploration and the ensuing mental processes going on in the physical motion. And so I'm going to use the fact that there are areas CA3 and the CA1 in the hippocampus, and in one of the areas there are actually connections in the neurobiology between the excitatory neurons. In the other area there aren't. And I put in both areas into this model now, and both areas have place cells in them. That's known experimentally. It's also known experimentally that these neurons drive those. I'm going to describe how it is having two kinds of place cells, some of which have feedback between them and enable you to do mental exploration, and some of which don't have feedback between them. How having these two kinds of things together enables you to do motor control, enables you to have a trajectory which actually follows along the activity which is described by the mental exploration here. Motor control. A rat has very, very complicated motor control. I don't want motor control to be the centerpiece of what I'm going to talk about, so I'm going to take the simplest possible motor controller that works and just show how there's enough information to drive the simplest possible motor controller. Make a better motor controller, and I'm sure you can do a better job. But the simplest motor controller you could possibly use is one that has an input, and there's a slow excitatory pathway, and a fast inhibitory pathway, and they're balanced, and so you get an output from this which is basically tells you what is the derivative of the input rather than what is the input. And now controllers of this system are common in bacteria. Bacteria basically find their way by swimming uphill anytime they're getting a better sensory signal. And if they're having a poorer sensory signal, a poorer signal toward an attractant, then they turn and go in some other direction. And that's an adequate, that kind of thing was discussed for many years now in the bacterial literature. Howard Berg has done elegant experiments on it. But you can use that same kind of controller for taking what would have been a random walk kind of motion, just moving around. And if you now have a sensor which says, are you getting better or are you getting worse? You can have an attractant over here, and once you're close enough to the attractant, you have a signal which is meaningful. The system of this sort will control you in such a way that you stay in the vicinity of the attractant source. The attractant dies off very rapidly with location here. All I'm showing you is that a very simple controller working on just taking a derivative and making you simply change directions if things are going badly is sufficient to be able to locate and stay near a nice attractant. Now you realize that you can do this in a more complex fashion. If you have the attractant slowly move, then the place that the animal would be located would slowly move. And so this kind of system is the basis for being able to follow mental exploration. What you have to do now is to make a motor system which sees the location of the mental exploration bump moving along as the location of the attractant, and the motor system will then try to keep you where that bump is. And a simple way of doing that is now to have the system run in the following way. Here's an area with feedback. This area does mental exploration. This area has learned what the proper trajectory is and is now about to recapitulate that trajectory by having a bump of activity move along through the trajectory to the goal. Here's this other set of place cells, which gets sensory input. It also gets input from this area. Now you notice that there are two bumps of activity present for these place cells. One is a bump of activity coming from the place cell information, and the other is a bump of activity coming from the mental side. And when these correspond, you get a big signal coming out here. When these don't correspond so well, you get a smaller signal. So you can see that this will result in a signal to the motor controller such that the motor controller will try to make these two bumps correspond. We'll try to put the animal at the location, at the physical location that corresponds to the mental exploration bump as it moves along. Which is that is true and undoubtedly an oversimplification. You actually will love to have some feedback which tells you run the dynamics faster or slow the dynamics down because you're not able to follow. But this one is putting it entirely on the dynamics of the mental exploration scale. That's right. But it doesn't work very well. But it does work, and you can see that I take the trajectory which I'd learned before and recapitulate it mentally. The thinner line shows the physical location of the animal with this very poor motor control system, but following the bump along. So you can see that you can actually manage to get the animal to physically move in a way which follows the bump. One of the interesting things for this is you've learned place cells for different environments, both here and here. When this corresponds to this, you get bumps which add up and so on, and the motor control system works. If the mental system thinks it's in one environment, but the place cell information coming in here from the sensory is in another environment, then these things don't add up. There's just no signal which comes out here because there's no correspondence. There's no way of getting correspondence between these bumps. One is a bump, one isn't a bump. And you suddenly realize what happens when you walk downstairs and say, how did I get here? Why am I here, etc. You know, if your physical location and your mental state location do not agree, then this system just collapses. And I understand why I was standing in the middle of the kitchen without any idea of what I'm supposed to be doing. What do I? Now, I'm going to just show you a movie for a second. The red dot represents the location of an animal. The black dots represent positions of neurons which are active at the moment. There's a sprinkling of black dots which are possible everywhere here. So in any frame of the movie, there'll be 6 to 10 neurons active as black dots. And as the animal moves around, the black dots will move around, of course. And what do I want to do? This is the kind of relationship you might get between cells being active and the position of the animal. This comes from a simulation I've done. And any neurobiologist, this would be a behaving animal, any neurobiologist would tell you, well, that's the kind of thing you expect to get. The activity of the place cells corresponds essentially to where the animal is. But in this case, causation is actually reversed. There's a bump of activity which is moving around spontaneously, and the animal is running its feet, it's doing its best to keep its physical location where the activity is. And so thus there are circumstances under which the direction, I would believe that the direction of the causality is going to appear to be reversed. That is to say, rather than the animal getting here first and then the activity pattern following, the activity pattern is going to lead and the animal is going to be following. You have to have good resolution of these things in time and space to tell it. But I think that that's one of the important experiments you can do with respect to whether the hippocampus is actually just responding to where you are, or whether the hippocampus is actually, through mental exploration, leading where you are.
We've done the same experiment. Suppose in some way I could record enough neurons in CA3 to pick up all the activity.
Right.
And then you're saying that I have the pattern as a function of time, and now what I want to do is find the pattern that happened at some point in the immediate past, and sort of impose that pattern in the immediate future, and then in a sense the animal should turn around. I should be able to make the animal go backwards by recording a activity had in the past, and just replaying it backwards, which would basically make the animal run a cycle in reverse. Is that the flavor? Now I'm imposing a memory.
I'd have to think about that. I'm really my thinking so far has really been in the forward direction. The question is whether the location which the hippocampus is reporting leads in time the animal, or whether the location the hippocampus is reporting lags the animal motion.
No. It could lead the animal in time, but I've just recorded, say, the activity of all the neurons.
Right.
So I have the past, I have this sort of pattern of activity in the past. And I just flip this in time. So now
if you flip it in time and start the animal from the appropriate end, it should lead the animal to go backward.
That would be, that's sort of the point of the experiment. I need to record from enough cells, know which ones to find a legitimate pattern, and then try to impose that pattern back onto the CA3. Trying this sort of experiment.
Yeah, well, as I say, the simplest experiment, as far as I can see, is the question of the lag lead. But that's not, lag lead is not really awfully definitive. And it would be very nice if you could do that one that way. If you could superpose a moving bump of any sort in an environment and do it in CA3 and then see whether you could lead the animal just through any path. It wouldn't have to be a path that you had taken before.
I see. So just, okay, so that's
Yeah.
crazy.
Yeah. That's right. And in the simple model here, one of them could be thought of as being CA3 and the other could be thought of as being CA1. One of them needs the intra-areal connections and the other one does not. Whether this whole thing actually is most logically mapped onto the hippocampus or some other brain area. And so many of the motions which people try to train to do in tasks, they think of the task as being basically a reflex action by the time they're done, but actually it is the intent to make some kind of motion in the physical world, which is what is learned, which is what Hebb was really going on about.
That you can do. With a virtual environment. Yes, you have the mouse on a ball with a virtual environment display, and then you just change let's say you have the rat is running straight ahead at a certain velocity, you can update the visual display at a different velocity. So you can perturb the environment in a way that doesn't match with what the rest with what the normal system is. Well, you can dissociate it. I mean, that's a way of dissociating the two,
Yeah.
which might be able to be good.
Let me just go to a little thing. Oh, this was just a particular example, of a particular environment and so on, where the model rat was started here and was looking for water, which is located in that location. And what did the exploration actually look like, and what did the synaptic change look like, and so on. Well, the first thing the rat did was to explore down this way, and then got stuck. Got stuck means there was no place for this activity pattern to go except backward on itself where it was already adapted out, and so it simply died out. Meanwhile, back here, there's still sensory information saying the rat's really here, so the activity pattern blob reinitiated there. One of the interesting things about mental exploration is, unlike physical exploration where I have to go back, in mental exploration, if you reach a dead end, you can jump. So the rat jumped back here, and now launched another mental exploration. The mental exploration this time went up this way, didn't go down this way because these have already freshly adapted. And finally, on about the fourth try, it actually went up and went this way and found water. If you then reinforce that pathway, the physical motion follows the mental motion up along the track. One of the interesting things. If you have a successful mental exploration and you lay down the synapses to do that, they're the same synapses to go back. So you would be able to retrace your steps. But if you have a non-successful one and just don't want to remember anything, you don't have to retrace your steps back. You can just say, let's reinitiate. All right, I could do a couple other things, but no, no. All right, the one last thing I want to come back to is this issue brought up by Boris about can you do parallel mental exploration? Yeah. Philippe Ponulac has been working with me some on ideas that he's had about how you might actually be able to explore parallel things at once. The system I've been describing to you so far has had this bump of activity moving around, and that's a single bump, and you can only explore a single thing at once. He thought about the question of can you actually have a wavefront which goes out from a point based on spiking neurons. The wavefront is a propagating wavefront of spike activity and can therefore manage to explore multiple things at once, multiple possibilities, rather than just one. And the answer is, you probably can. The answer is, on the other hand, at present neurobiology does not really suggest that that the rat can do that, but that doesn't mean that other animals can't do it or the rat can't do it in some other particular paradigm. This is the nearest experiment I can see of real modern neurobiology to anything which looks sort of like mental exploration. These experiments from Matt Wilson's laboratory. The rat is coming along here, and either it's going to take the blue trajectory or the red trajectory. There's actually a reward up there. No, I'm sorry. There's a reward down there. That's the dead end. And the rat has learned the system many, many times. This is the cell position along this track, this linear track, as viewed by the activity of a substantial number of hippocampal place cells. It's not frightfully well defined, but you can see the activity of the place cells. Here the animal is stationary. What the animal is doing is activating some cells which correspond to taking the blue path, activating cells corresponding to the blue path. A little later it starts activating cells corresponding to taking the red path, and then after a while it actually starts making a trajectory which follows the red path. This looks like sounding when one is sounding and repeating, testing, and after a while making a decision and actually then having an activity pattern which follows along with the way the exploration has gone. Now this looks somewhat like what I've been telling you. It's not exactly like it. It involves spikes in a way which is more detailed than the way that Ponulac has. It also does not succeed in exploring two things at once. It only explores one thing at a time. But it's this kind of thing moved on to action potentials, which actually may succeed in doing what Boris would like, exploring more than one thing at a time, and maybe part of how we get our exploratory power. Yeah. For real place cells, first of all, when the rat is sitting still, the place cells where the rat is tend not to fire. And what this is it's simply the activity of cells which would be active when this. What does this red dot mean? That's a cell whose place field center is located about at this point, and that red dot means simply that that cell fired. Place cells are defined by mean activity, as the rat was wandered around over lots of things. And then we've just done this plot. And you could say that this cell temporarily has a place field which corresponds to where the rat is. But I don't think that would be I don't think there's anything to support that idea. Well, no, but the other alternative is that there's been, in some sense, a deliberate mental exploration. And if you say only place cells fire because of where you are, then there's no way you can embed mental exploration into place cells.
They fire most of the rest. But that doesn't mean they don't fire at all.
They fire when the rat is in that position and actively exploring. That's the other thing which controls when place cells fire.
I think that I think what you're asking is, so when you're recording from these neurons or exploring the environment to define place field and information levels, the neurons are noisy, right? And they might fire actively and will not. Yeah, so, right. So empirically what that means is that the firing rate then goes to 0 regardless of
And place cells, as defined, are wicked averages over things which shouldn't necessarily be averaged over. There's a subject of overdispersion, which in my view is interesting because it relates to how you might unscramble the world into objects. But all this, what the take-home of this is, here are two different trajectories of a rat wandering through. What is shown in gray is the place field of a particular cell. This is the place field as determined by the usual kinds of its behavioral measurements. Now, in one of these trajectories, the rat actually went through and fired 66 action potentials. In the other trajectory, went through and fired 0 action potentials. You can figure out how much they should have fired. That shows the probability distributions of firing you would have expected. These things are so many sigma off scale, it doesn't count. And you can see that there are things which control place cell firing which go beyond just the question of where you are. And in this particular case, they're associating it with attention. And I think it's also associated in some sense with objects, but that's another subject that I'm not going to get into. Okay, bottom line, just very quickly. I think that you can see, as I've been discussing the subject for the past 40 minutes, what's important is the collective aspects of the activity. And in order to describe what's going on, you need to understand how that collective activity evolves in time. It's the collective dynamics which really represents why the system has robust behavior. And I think that one of the focuses of neurobiology has to be on trying to use multi-electrode recordings, trying to use the modern abilities to go in and excite big collections of cells to try to get one's teeth into the collective variables of neurobiology. Yeah, and it isn't that the cells are all doing the same thing. You see, what's interesting to discuss is the motion of this bump. Now, there's no cell that corresponds to where the motion of this bump. This bump, it's like sound waves. You know, it rises above the details of what's going on underneath. You need to be able to describe the motion of the bump. You need to be able to have equations which describe the dynamics of the bump, and the fact that there are multiple sheets on which this bump could exist and it could jump from one to another, without getting down to the nitty-gritty of what all the neurons are doing all the time, which is just too far down. And you need to try to make this connection in the way that you make the connection between the laws of hydrodynamics and molecular collisions. But that's my current zeal, my current religion.
I'm curious, do you think there's an intuition that during mental exploration, the neural representation into the public is essentially the same as during more active behavior? So where does this intuition come from?
Say that again.
You somehow, if I'm correctly, assume that the neural representation is similar during mental exploration and during active behavior.
Yeah.
I'm curious where this is, for me, it's counterintuitive.
I didn't have an alternative hypothesis, and if you leave it the same, I said, can you actually make a system which is capable of doing functional mental exploration, from mental exploration of a known environment to physical motion? And can you do it with things which look somewhat like what neurobiology has to offer, without adding too many things which I don't know that neurobiology has to offer? That was the restricted domain in which I did my thinking.
So basically most of the data comes from active exploration, as you said. So that would bias you towards assuming
It does not come from purposeful exploration. It doesn't come from trying to find water in a known environment and so on. It simply comes from wandering around, and the only purpose of the wandering around is to get a food pellet which has been dropped at an arbitrary location. So I think it's really, the environment is really learned through physical exploration, and there are no mental exploration aspects important in establishing the place cells, is the position which I've taken.